Thank you all for coming to another of the plant health series from the Royal Society of Biology. Today's webinar is quite an exciting topic and one that I'm particularly interested in because it's a plant pathology topic and it's being presented to us by an esteemed collaborator and colleague and I suppose mentor in some ways of mine, Dr. Kendra Baumgartner, who is a research plant pathologist for the USDA-Agricultural Research Service. And she's based at the University of California, Davis. She's a specialist in fruit and nut crops, particularly grapevine. And her skill set is quite far-reaching, encompassing molecular, basic, and applied plant pathology. So she's come through a background from forestry in Syracuse in New York, and then her PhD focused on Armillaria root disease in grapevines and forests in California. And then she joined the USDA. Through her career so far, she's worked in quite a number of collaborations, both within the USA and internationally, including receiving a fellowship from the Organization of Economic Cooperation and Development to come over to the UK and work with Bristol. And also I've sort of come over to work with her in her lab. So that's an association with the Royal Horticultural Society now as well. She's also led a team of 8 labs in the USA working together on trunk diseases of grapes, pistachios and almonds, funded by a grant from the USDA Specialty Crop Research Initiative. Her current research focus encompasses Armillaria mellea, as well as Eutypolata, Phomopsis viticola and various species of the BOTRYOSPHAERIA. She also conducts extension work through the Californian, what's it called? Californian, it's a research extension kind of operation. Sorry, Kendra, I've lost those words. It's the University of California Cooperative Extension Network, and is an extensive author of many different scientific articles, over 70 articles and 10 book chapters. So she's really accomplished a lot in her time, and she has an upcoming book chapter coming out if you're particularly interested in grapevine diseases, and which she's senior editor of. So keep your eyes peeled for that, and I will hand over to Kendra to really take the floor and show you what she's all about. So thank you, Kendra.
Thank you. Can you hear me all right? All right. Thanks so much, Jassy, for that excellent introduction. And it's great to see you again. For those of you who don't know me, I'm a plant pathologist, like Jassy said. And we are both kind of mutual world travelers who've been across the pond, so to speak, working in each other's countries over the years. Let me start sharing my screen here so you can see my presentation, and I'll start speaking. All right. Well, I've got my timer going too. So today, as you can see, I'm going to talk about grapevine diseases and diseases of tree crops in California. There will be 2 parts to my talk. The first will be on Armillaria root disease of almond, and the second will be on ESCA of grapevine. And ESCA is a grapevine trunk disease. Both of these diseases are found worldwide in every major growing region for these 2 crops, but I'll be focusing on the work I have going on in California specifically. So before I start speaking about almonds, I wanted to share some of the statistics on just how important almonds are as a crop in the United States and especially in California. You can see in this table almond production, and I've listed almonds and all of the different stone fruits. In parentheses are the US states that they're grown in primarily. And then this column on the right shows the acreage from the last available numbers, which were compiled in 2021 by the USDA National Agricultural Statistics Service, another branch of the USDA. And you can see I've converted these to hectares, so they're familiar to you. But almonds are exclusively grown in California in the United States, and they far exceed acreage for all of our other prunus species, which are stone fruits. I just also want to mention that a few of these on here, peaches are grown primarily in California, but also in some other states in the US. The Rocky Mountain state of Colorado, you know, you might think of snow and skiing. That doesn't really jibe with peaches. Well, there's parts of Colorado where they can grow fruit crops. Where they have enough chilling hours, but it's warm enough to bring them to ripening. But we also have production of peaches in the southeastern US states of South Carolina and Georgia. And then I want to point out here, tart cherries are grown exclusively in the northern US state of Michigan. And I'll be talking a little bit about those today. Okay, so for those of you who aren't familiar with Armillaria root disease, let me just give you kind of a crash course in disease epidemiology here. This is a disease caused by a fungus called Armillaria mellea. It's a basidiomycete fungus, and it's a chronic disease problem that's present on a site before the orchard is even established. The fungus has a very broad host range of hundreds of plant species, which includes native California tree species. When those trees are cut down and planted hosts are put in, like this walnut tree, which has been knocked over by a tractor, these trees get infected by growing into direct contact with residual roots, which are woody roots that were infected that remain in the soil after infected plants are cleared. And these white bits are the vegetative stage of the fungus, technically known as the mycelium. For Armillaria, we call these mycelial fans because they develop kind of a fan shape where they form beneath the bark and between the bark and the primary xylem. Armillaria is not a soil-borne fungus, it's a wood decay fungus. So it doesn't have a free living stage that can survive in the soil. It has to be in woody roots that are buried underground. And you rarely see this fungus grow in the trunk of the plant above the soil line. It's typically restricted to the base of the trunk and the woody roots. Here are the lovely mushrooms of the fungus. Well, lovely for me because I'm a mycologist, not lovely for dying trees, of course. This fungus makes wind-blown spores. These spores are not thought to be a significant means of colonizing new trees within the lifespan of a typical orchard, though, in California. The fungus also makes some very unique structures called rhizomorphs. That's what these black root-like structures are. Inside, they have actively growing fungal mycelium. Those white mycelial fans, they're kind of repackaged in these rhizomorphs, and these grow out from infected roots. They're quite small for Armillaria mellea, which is our most widespread and our most aggressive Armillaria species in California. Most of the ways that, the main way that planted hosts get infected is by their roots growing down into the soil into direct contact with residual roots, rather than rhizomorphs growing out from infected roots. Okay, this is an overhead view of an almond orchard. This picture was taken years ago, but the results still apply. Almond is very susceptible to Armillaria root disease, mainly because it's grafted onto rootstocks that have a peach background, and peach is super susceptible compared to almond. The grower at this site had replanted what we call a disease center, which I've circled here in the center, where almond trees have been dying from Armillaria, replanted them with walnut, which tends to be a bit more resistant to this disease. Because the fungus has such a broad host range, it's hard to, so to speak, rotate different crops into an area. That said, some are more resistant to the disease than others. Here's a peach orchard from kind of the ground view rather than the bird's eye view I showed you before. This is a peach orchard. I've circled the big Armillaria root disease center in it. And a colleague of mine, Roger Duncan, who's the University of California Cooperative Extension Farm Advisor, invited me to this site several years ago. And the grower had kindly let Roger use this as an opportunity to set up an experiment. And so in this site, they had Armillaria root disease that killed off many of these peaches. Peach is one of the most susceptible crops to Armillaria root disease in the world. Also at this site, though, were some other root diseases, and I've listed them here. I have to give credit to other root diseases. Armillaria is not the only important one for peach and almond. We also have the ring nematode, which in a sort of disease complex with the bacterial canker pathogen Pseudomonas syringae causes a disease on peach that we call peach tree shortlife. We also have other nematodes, root knot nematode, lesion nematode. Peaches are susceptible to Phytophthora root disease, which is caused by an oomycete filamentous microbe, not a true fungus, but an oomycete rather. And they're susceptible to crown gall, which is caused by the bacterium Agrobacterium. At this specific orchard, Armillaria root disease was a problem, but peach tree shortlife was also a problem too. Roger, my colleague, took advantage of this opportunity to plant some experiments, and he put in experimental rows of peaches grafted onto different rootstocks. This rootstock is called Nemigard. Obviously, it didn't do well and is susceptible. This rootstock, Viking, it's called, the peaches did much better on it. And so then Roger called me up and said, well, I'm not sure if this rootstock, Viking in particular, is resistant to either Armillaria or peach tree shortlife. Can you help me with the Armillaria question? And so we did. We've been screening, we developed a screening assay for Armillaria. And without going into a long explanation of why we used this approach, I'll just briefly say, as a pathologist, I would always prefer to try to inoculate plants and evaluate their susceptibility in the greenhouse in potted plants. But for Armillaria, for whatever reason, it's not easy to do that. It's not easy to recreate what happens with infection in the field with the woody roots being infected in a potted plant in the greenhouse. And so instead, I developed this assay where we root plants in a tissue culture medium, which is agar-based. Maybe you can kind of see here the gel at the base of this box that the plant is in. We grow the fungus separately for 2 weeks in a liquid culture and shake it to oxygenate the medium. And these little balls here are of the mycelium of the fungus, but they kind of form, they get round. We grind those up with a handheld blender, and then we pipette them onto the surface of the medium. And after another week, it grows to colonize the surface of the medium, and it colonizes the entire medium. So no root escapes the fungus in this assay. It's a very extreme assay. By week 8, we can pull roots from that medium, stain them, look at them under the microscope, and see the fungus colonizing them. And then by week 10, some of the most susceptible rootstocks start to die. At week 12, we count up who's dead, who's alive, and our mortality rate is our measure of resistance. And colleagues of mine have used this assay also to look at resistance in pear. I've used this assay for grapevines, almond rootstocks, walnut rootstocks, and then colleagues of mine have worked with this on pear as well, and some other horticultural crops too. I'm going to talk a little bit about the rootstocks for people who are interested in the plant breeding side of things. The information I'm going to present now comes from a publication called Almond Rootstocks, which is by my colleague Roger Duncan, and Joe and Kathy are both also farm advisors in California who focus on almonds. But this table here, I've lifted bits of the table from the Almond Rootstock publication showing the Nemigard rootstock that I was just showing you a picture of, where the peach plants were very, very much failing. We've screened this rootstock using our screening assay, and it turns out it is indeed susceptible to Armillaria root disease. This is a rootstock that comes exclusively from a peach background. All of its parents are peaches. Almonds and peaches within the prunus are relatively closely related, and so there's a lot of good graft compatibility between them typically. All of these other peach rootstocks, Guardian and Lovell, they're also very susceptible to Armillaria root disease. These are still the most widely planted rootstocks for peach and also almond in California. Unfortunately, also this set of peach almond hybrids, those that I've screened, Brights 5 and Hansen 536, these two are also susceptible to Armillaria. but there is hope. As I showed you before, with the picture of the Viking, the plants on the Viking rootstock, we have these Viking, which turns out to be resistant in my assay. But I also want to mention a whole set of plum hybrids, which include, when I say plum, I mean plum in the broad sense. These are very distant relatives of the cultivative plum, which is Prunus salicina. These are plums you've never eaten these things. They're wild relatives of plum. Saricifera is something, though, that all of these plum hybrids have in common. And two of those that I've screened to date, Krinsk 86 and Mariana 2624, are resistant to Armillaria root disease. And there's interest among people who breed for rootstocks for both peach and almond to find ones with this Prunus saricifera in it, because we think this is an important source of resistance. And I've also listed some of these other wild plum relatives that are part of the background, the genetic backgrounds. I'm part of a new project, well, over the past 2 years now, not so new, breeding rootstocks for resistance to Armillaria root disease for Prunus. Here I'm on the West Coast. Here's California with a little peach and an almond, and I work very closely with a peach and almond breeder, Tom Graziel, on our part of the project, focusing on breeding new Armillaria resistant rootstocks for almond. We work with a big group of people on the East Coast of the United States, though, and many of them are located at Clemson University, which is in the US state of South Carolina. Ksenia Gasic, who's listed on the top here, she's a peach breeder at Clemson. We also have colleagues in Georgia. South Carolina and Georgia are important peach producing states. I want to mention we have a group of people working with us on tart cherry in the US state of Michigan at Michigan State University. Cherry is too distantly related from peach and almond to share rootstocks. Nonetheless, we share a lot of our laboratory techniques, namely methodology involving infection assays, and also fungal strains. Tom Graziel does the breeding, as I said earlier, with Prunus saricifera, our plum relative. And the way he does this is he collects pollen from almonds. He also collects pollen from certain peaches in his breeding collection. And the way he crosses these with the Prunus saricifera mother trees, so to speak, is to bag the entire tree. And inside there, he releases the pollen to when the flowers are developed on the mother tree. And to help improve pollination, he puts bees in there, and he doesn't just use any old bees. Honey bees are kind of fickle when it comes to our weather conditions during bloom. Sometimes they don't like the bad weather, well, bad under California standards. And so Tom uses bumblebees. And bumblebees will fly in windy, rainy weather, apparently. And this increases the chances of pollination of the flowers. And he puts, I've circled here a tube where the bees, this is the only place they can kind of get in and out of that box, which has their hive, so to speak. And he encircles that with pollen. So when they come out of that tube, they've got pollen all over them. And then hopefully they will visit the flowers on that tree and create new crosses for him. And then we work with the folks at Clemson to get those, germinate those seedlings and to genotype them, and then to start screening them in our assays. They've also modified the in vitro assay, so to speak, to accommodate some rootstocks and some experimental accessions, germplasm accessions that do not like rooting in an agar-based medium. And they use this rooting medium called a phenolic foam. They pour into it a nutrient solution of macro and micronutrients, and these support both the roots of the plant, and this fuzzy white stuff here is the pathogen. The roots also develop a little bit more of a similar in appearance to those that you might see growing in potted soil-based media. And here they've used this assay to to successfully compare susceptible rootstocks like Guardian, which is a pure peach background, to here's one of the resistant plum hybrids, which is an open pollinated seedling of Prunus cercifera, which is doing much better under Armillaria infection than is the susceptible Guardian. So now I'd like to switch gears and talk about grapevine trunk diseases. I want to first mention that we do a lot of our research in table grape vineyards in California. These are mostly located in the southern San Joaquin Valley. It's south of the capital of Sacramento, but not all the way into southern, what we call Southern California, which is like the Los Angeles area, which is vast, sprawling metropolis. This is north of there even. Table grapes, I want to mention, they're the same genus, plant genus and species as wine grapes. They're Vitis vinifera. However, they're seedless. And to get their seedless trait, they all share one parent in common called Thompson seedless. And Thompson seedless, that's how they get their seedless trait. You know, these are seedless because we want to eat them fresh. We don't want to bite down into a seed. We want no seed in there. Thompson seedless tends to be quite susceptible to many of our main trunk diseases in California. And even though these table grapes are grown in a region that is very dry, you know, at most there might be 10 centimeters of rain per year, the high level of susceptibility brings about a relative, more disease than you would anticipate given the lack of rain. Okay, and I wanted to share some statistics on grapevine production in the US with you really quickly. I'm sorry I didn't convert these to hectares. These are in acres, but you can see the relative differences. In California, we have, right now we have about 800,000 acres of grapes. The majority of those are wine grapes, but we also have a fair amount of raisin and table grape acreage, which is, like I said, located in the southern San Joaquin Valley. Washington State, which is 2 states north of us, is the second largest grape producing state in the United States. Pales in comparison in total acreage, but it's still important. I want to mention here, they grow not only wine grapes, many of which are the same cultivars which we grow here in California, which are French cultivars, but they also grow juice grapes. And the juice grape that they grow is Vitis labrusca. It's not Vitis vinifera at all, and the cultivar is mainly Concord. This is the juice and preserves grape. That's very sweet. That's grown in much of Washington State. Okay, so I'll talk about a grapevine trunk disease called ESCA. It's found in every major grape producing region of the world, not just in California. It's a fungal disease of the wood, but it causes these very striking leaf symptoms, which involve scorching around the edge of the leaf and in between the veins, and also a discoloration, which can be yellow, as shown here for Sauvignon Blanc, sometimes yellow and red, as shown for Chardonnay, sometimes just yellow, as for Grenache. I want to mention quickly that Sauvignon Blanc is one of the most susceptible cultivars to ESCA in the world. We have other trunk diseases in California too, which I'll just mention briefly. One of them is called BOTRYOSPHAERIA dieback. And the dieback type symptom involves a dieback of the shoots. And I just kind of wanted to walk you through how this series of events happens over the course of a year. What happens first is a shoot will grow out of an infected fruiting position. We call this fruiting position here a spur. These are spurs along a cordon or an arm. And these fungi infect the pruning wounds during the dormant season of the grapevine. And so a shoot will grow out from an affected spur. It will never quite catch up to neighbors on uninfected spurs, possibly on the same cordon. In midsummer, that shoot will often die. If you cut through that spur or even that cordon, you may find a wood canker. And this is one of the wood symptoms of BOTRYOSPHAERIA dieback. This brown part is totally dead. It's not useful for the plant. It's not even useful for the fungus. The fungus survives kind of in the margins of this canker. And eventually that spur will die off completely. The next spring, no shoots will grow from it at all. And vines can accumulate these infections over time in different spurs during different years. You know, it's a perennial crop, and the woody permanent portion of the vine accumulates infections from BOTRYOSPHAERIA, DIEBACK, ESCA, and other trunk diseases. And eventually, the more spurs that die, there's fewer shoots, and the grower ends up having a very unproductive vineyard. The vines are still alive, but they're just not producing enough fruit to make it profitable, and they have to be removed. And so this is the damage that trunk diseases do, primarily the dieback type diseases do to California vineyards. They lengthen the productive lifespan of the vineyard. ESCA is different though, but first I would just want to mention what these fungi do to the wood. They're all wood infecting fungi. They're unrelated genetically, so the ones that cause the different diseases, and I'll show their scientific names in a moment, but what they share in common is they colonize the woody cells and different cell types. The infections tend to be within centimeters of the entry point, which is the wound that the fungi get into in the first place. The discolored wood, you can't see, like the wood canker I showed you earlier. The ESCA pathogens cause these black spots in concentric circles, and also one of the fungi that causes ESCA causes a white rot type of wood decay. You can't see these symptoms unless you physically cut through the wood. Nonetheless, even though these fungi live internally in the wood, they eventually make their way out to the bark surface where they sporulate and spread to nearby vines. There's no chemical known to mankind that you can spray on the outside of the vine that gets taken up into the wood to kill off these fungi. They're chronic infections. And so the best approach is to prevent the infections from happening through the pruning wounds in the first place. I've listed our 4 main trunk diseases in California: BOTRYOSPHAERIA, DIEBACK, Eutypa DIEBACK, Phomopsis DIEBACK, and ESCA. BOTRYOSPHAERIA, Eutypa, and Phomopsis, the names all come from the scientific names of the pathogens. ESCA is quite unique in that these 3 fungi that I've listed here, they're unrelated completely, yet they can cause the same symptoms. And often we find them in different combinations in infected vines. And here's just a point, what we call those as plant pathologists are mixed infections. You can find multiple pathogens within the same vineyard and also even in the same vine, even in some pieces of wood. This is a sample with a Petri plate with 2 of the ESCA pathogens growing out of some of the same bits of wood. You know, and you have to think, these are microbes, you know, they're microns in size. When we plate out a piece of wood, you know, it's like a planet for them, so to speak. So it's all relative here. But what's interesting about these pathogens is that they're unrelated and can cause similar symptoms, either together or separately in the wood. We as a lab work very closely with diagnosticians at private diagnostic labs and also at state and local public labs to develop diagnostic tools to identify these species to the species level. Unless you're a trained mycologist, it's very difficult to discriminate these fungi in culture. And so we work hard to develop reliable, rapid PCR-based or rather DNA-based methods. These are a list of the species that we in our own lab have identified to the species level and described them. They're new species known to science to cause trunk diseases. And it's a long list we've gathered up over the years. They're not all found in California. Some of them we've described from vineyards in Texas, in New York State, in Washington State also, from our different surveys of vineyards with trunk diseases. So really quickly, I want to mention some of the work we've done with ESCA. ESCA causes not just these very striking, scorching symptoms on the leaves, but the disease also damages the fruit and disrupts the ripening process. It causes spots on the fruit in its most mild form, and those spots are not the fungus. We're not really sure why they form, but it's a symptom of the disease. It's not a sign of the pathogen. And when you get so many spots, the fruit actually looks like a red grape. And this is Sauvignon Blanc. It's supposed to be a white grape. You have one here at the left of your screen, which is a healthy cluster. We can get healthy and symptomatic clusters on the same vine. It's all dependent on where that shoot, the shoot from which they grow, is connected in the vascular system to where the fungus resides deep inside the woody tissues. And so often what growers will do in vineyards with ESCA is before harvest, they'll go through and physically cut out the symptomatic clusters. So those don't get harvested in the mix with the healthy clusters. But there isn't really a clear understanding of exactly how bad these spotted clusters affect wine quality. And so I've been working with a wine chemist. Her name is Erin Rumbaugh. We just hired her to work on primarily on how smoke from wildfires affects grapevines, but she's a chemist, and so she's been working with me on this research question as well. I just want to mention too, if I didn't make it clear, these fungi spread by spores that are, depending on the species, it happens by either the wind or the rain. They originate from grapevines, obviously, but some of these species also attack tree crops and native trees, and then they can contaminate nursery stock as well. I also want to mention something interesting about the disease cycle is that the pruning wounds, depending on when we prune the vines during our dormant season in California, the period of susceptibility can vary. So if pruning wounds are made, or vines are pruned rather in December, those wounds can be susceptible for 3 to 4 weeks. If the vines are pruned in March, closer to bud break, they can be susceptible for only a matter of days. So just an interesting bit of information about the spread of these diseases. And thus what most growers do is we recommend do not prune in the rain because rain brings out the spores. And then fungicide applications can be fairly effective if applied before the next rain and after pruning. One thing that happens, vines get infected, the fungicides are not 100% effective. And one technique that growers use is called trunk renewal. It involves cutting away the entire top of the vine where all of the infections are localized because they've been getting in through pruning wounds over the years, and carrying that out of the vineyard and retraining an entire new vine from a new shoot that forms at the base of the trunk. A grower who I work with on this technique, he calls it decapitation because you're effectively cutting the head off the vine. But it's a drastic approach because you have to then retrain a vine, which can take 2 years to get a full canopy producing again. But it promises to get rid of all the infections. And we worked through an experiment with a grower with a Sauvignon Blanc vineyard testing this approach. This was a vineyard that had very serious ESCA. And we worked with the grower to identify the symptomatic vines in October of 2017. Then in February of 2018, we came in and used a chainsaw to cut these vines. This is above the graft union, mind you. These are all grafted onto rootstocks. We treated those wounds with fungicide and then retrained new shoots from the base of the trunk into a new canopy. And this is one growing season's worth of growth on one of those decapitated vines. The advantage of this approach over replanting the vine completely is that you can utilize a fully developed root system, and that can more rapidly grow shoots into a full vine rather than a replant, which is a much smaller little vine, which tends to have a tough time getting established in a mature vineyard. And then when we came back, this just shows a picture of one of the vines in 2020. But when we tallied these vines, these are numbers of the original 97 decapitations they went through in 2018. These are numbers of who all survived. And these come from just this past growing season of 2022. So 73 of those 97 vines, we retrained into vines with no symptoms of ESCA. 23 of them had to be replanted completely. Either they died for some other reason, or a frost, for example, or the shoot never formed at the base of them. And then one of those vines does have symptoms of ESCA. One of the things we wanted to know is if this technique worked to retain fruit quality for making the wine. Because that's what matters. You know, it doesn't matter that you've got a nice, healthy looking plant. What matters is the fruit for making Sauvignon Blanc. So in working with Aaron Rumbaugh, my colleague, a wine chemist, this is kind of a complicated slide, and it's my last one, so bear with me here. We collected fruit from 4 different sets of vines throughout the vineyard. We had healthy vines that we collected healthy fruit from. We had those retrained or decapitated vines, so to speak. We collected their healthy fruit from. We had mature symptomatic vines in the vineyard that had ESCA, which were never decapitated, which had both healthy fruit and symptomatic fruit on them. And so we collected all of these, and Aaron peeled the skins away from the fruit and analyzed in the skins the tannins, which are chemicals that are important for flavors in white wines in general, but particularly Sauvignon Blanc. And this figure shows here all of these dots are samples of sets of grapes from these 4 groups. The arrows originating out from the origins of the axes, as they get farther out, point to different chemicals different tannins that she identified. And the farther the closer to the head of the arrow are the samples that had the highest concentrations of those chemicals. These big circles that circle the samples from the same treatment group, so to speak, just kind of give you a better view of how they were all clustered together, so to speak. And you can see that the ones from the symptomatic fruit from the symptomatic vines, that the disease is really changing the tannin chemistry of the skins in that fruit, such that, and Aaron told me, I forget exactly which chemicals they are, but some of these tannins have been shown to be associated with green or vegetative flavors, grassy type flavors that persist even through the winemaking process. But what was interesting for us is that even these, this red group here, these are our healthy fruit from our retrained vines. It fortunately is near its friends, which is the healthy fruit from the healthy vines and also the healthy fruit from the symptomatic vines, but it's still a little bit different in its tannin makeup and its skins. And we think that that might be associated with the fact that even though the base of the vine is 20 years old, that the canopy of the vine is only several years old and it's still quite young. And fruit quality and the wine that's made from it can change over the course of a vineyard's life, and in complex ways. This is just one set of chemicals that are associated with grapevines, the skin tannins, but it's obviously an important area of research for wine chemists. And also, I just want to mention, it's a bit speculative to say that these chemicals would be the same in the finished wine. A lot can happen during the fermentation process. So you can't really always relate concentrations of chemicals in the juice of the fresh fruit to what happens in the wine, the finished wine. And so with that, I'd like to thank you so much for your attention. And thank you again for inviting me today. I'm going to stop sharing my screen so we can have a question and answer session.
Thank you so much, Kendra. Yes, if you've got questions, please put them in the Q&A box rather than the chat. I have got some questions already coming in, but I thought maybe I'd start with just that last story about the ESCA and that one, particularly that one plant that seemed to still regrow despite the decapitation. I wondered, with this fungus, does it translocate in the wood much? Like, is it because it was too far down the trunk and when it was decapitated it hadn't actually been shed? Or is it, still just like there's spores around and it's easy to be reinfected and it didn't really escape the rest of the inoculum in the environment?
Yeah, it's a good question. I mean, I would say that when we decapitated vines, we took wood samples of the trunk and noted whether there were wood symptoms or not. And then we brought them back to the lab to try to culture the ESCA pathogens from them. We had wood symptoms in a high proportion of them, but we identified the fungi in a very low proportion of them. And I don't know if it's because the fungus was unidentifiable, like was it just, they are slow growing pathogens in culture, and it can be difficult to culture them, especially if you don't grab the piece of wood that has the microscopic fungus in it. But also it's possible that these wood symptoms form quite distant from where the pathogen is. We know that these trunk pathogens are not, they're not in the fruit, they're not in the green shoots even. The fungi reside in the permanent woody structure of the vine. But what does get translocated out to the shoots potentially are phytotoxins, chemicals that these fungi make. And the ESCA pathogens have been shown to in vitro at least, make a series of toxic chemicals that are toxic to plant growth.
Yeah, that's really interesting, because there's another question about that wood, because obviously they do make spores, but is that wood and in the process of decapitating, creating sawdust, Adam asked if any of that sawdust can itself be a source of further infection?
It's a good question. I mean, certainly when inside the wood, the part of the fungus that resides there is the vegetative stage. Typically when these fungi sporulate, they do so on the bark surface. So it's difficult to find their fruiting structures. I mean, they make, you know, they're what I call the black dot fungi. You know, you might find a tiny kind of pen point sized dot on a vine, of which there are many if you've ever spent any time crawling around on your hands and knees in the vineyard looking at grapevine trunks. But yeah, it's difficult to know whether we're moving around the fungus, so to speak, by doing that. And there are certainly fungi that, you know, they make microscopic spores. So if you see dust blowing around in the vineyard, inevitably there are spores in there too. We do try to do the decapitation. We do it at the time of pruning because it is very difficult to tear those vines away from the trellis system when all of the canes are still on them. Yeah, after they've been pruned, it's easier to just go through and chainsaw them and decapitate them. But we do try to prevent any infections of trunk, the remaining part of the base of the trunk. We do put a protective coating of a fungicide on there, because that's a very large wound that could potentially get infected. Yes.
Yeah, sticking with ESCA, and then there's two more questions on ESCA for now. John asks whether you've done any spore trapping work to understand the seasonal timing of the trunk infecting pathogens, or is that already known? And
we've done lots of spore trapping.
Yes.
The ESCA pathogens, we don't have a lot of experience spore trapping them. We have more experience with the DIEBACK type diseases. I would say that the BOTRYOSPHAERIA and Eutypa diebacks tend to make spores with rains during the dormant season in California, which is typically between December and February or March. The Phomopsis dieback pathogens tend to start sporulating after bud break, so with rains that happen in March and April. And April is pretty much the end to our rainy season here in California. There's been a bit of work published in California on the ESCA pathogens, and they do tend to sporulate whenever there's any source of water. During our growing season in California, we do occasionally on very hot days, hot meaning like 40 C, it gets hot. Growers will circulate overhead sprinklers in the vineyard to cool the vines and allow the fruit to still ripen properly, so the vines don't suffer from heat waves. Spores can be dispersed during those times of the ESCA pathogens in particular. That's been found. So
Yeah, it's a difficult trade-off.
Yes,
Yeah.
yes. Although we make the assumption that the wounds heal faster when the vines are actively growing. So whatever wounds might be there that hadn't healed, maybe wounds that had been made through we have a joke, we call it tractor blight. When tractors drive through the vineyards, sometimes they cause gashes in the trunk. That would be a type of wound you would have during the growing season, because there's a lot of tractors going through the vineyard spraying for powdery mildew of grape. Primarily, that's one of our most important diseases, limiting grape production in California, at least. But that would be a wound, tractor blight wounds. Also, another wound that we get in California, we've had labor shortages going on seriously for about the past 6 or 8 years in the United States, but especially in California. Part of that, sadly, is because of savage drug wars going on in Mexico, and people are not alive or are just not migrating across the border legally or illegally. And so we have few laborers to do work. And so more and more vineyards are being mechanically harvested and mechanically pruned also. And the machinery can damage the cordons and the trunks, unfortunately. And that can cause wounds. So after harvest, you can get wounds from that.
Yeah. And then the last question on ESCO is about whether there's any biocontrol available.
There are some biological controls that are commercially available in the US now. We have fungus, and they've been tested primarily in Europe for many years, and they're relatively recent to the US market. Trichoderma species, which are an ascomycete fungus, that can be applied to pruning wounds to hopefully colonize the wounds before pathogen spores do. That's relatively effective, as long as they're applied before infection. So, and they have to be done annually. It's not something that we have a good understanding that lasts permanently. So,
yeah.
Even though trichoderma can colonize the internal parts of woody plants, not just grapevines.
Yeah. Yeah, that's interesting how trichoderma seems to get applied to so many different pathosystems.
Yes.
Yeah, I always wonder about the generalists versus the specialists, and how what's it going to work against. But yeah,
well, it's a mycoparasite, right? Like, and trichoderma can be very aggressive, even against Armillaria mellea. It eats Armillaria, and it does so for a broad range of fungi, and more aggressively so for some other than others. So.
Yeah, there was one question about Armillaria, about your agar-based method versus investing sterile soil. Doing that instead of the agar, but I guess you also had a third growing system, which was that I can't remember what you called it now.
The phenolic foam, yeah.
The phenolic foam. What are the sort of the kind of pitfalls and the pros and cons of the three? Like, in what circumstances do you use each of those systems?
I would say growing plants in pots in a soil-based or a soil-less medium definitely creates more field-like root systems and root structures. However, the way that we would recreate infection in a potted plant is by growing. The way we've done it is we grow the fungus on a Petri plate, and then we take sterile bits of wood that we've autoclaved, and we set it on top of the colonies, and then the fungus will colonize the wood. And then we bury that next to the roots of potted plants. And you can get some infection with that, but it seems like no matter how many bits of the fungus that I try to create, inoculum, so to speak, they don't survive. You don't get a lot of infection. The infection goes very slowly. And so rarely do the plants show symptoms or die. And so you need symptoms and or mortality to get a measure of resistance among maybe different sets of germplasm or rootstocks in our case that you might be testing. And that was the advantage of the agar-based assay, is no plant can escape the fungus. But the pitfall of the agar-based assay, there's two that I've encountered. One is not all plants want to make roots in that agar-based medium. Walnuts are particularly unhappy with that medium. And I would say the other problem is that it's a very severe screen to put the plants through, that even rootstocks we've seen to be resistant under natural conditions in the field, still a lot of them die in the agar-based approach. So that's the downfall with that. And that's the importance of having controls in our study, a resistant control and a susceptible control, along with our experimental rootstocks.
Yeah, I can definitely vouch for the difficulty of carrying out Armillaria pot trials. I think a statistic of 50% in your positive control with a highly susceptible host is considered good. Which, if you think about the replication numbers you need to show the effect of a treatment that might be quite subtle, that's a big footprint for an experiment and a long duration to see whether something eventually doesn't work. Yeah, so the Argo is definitely helping to accelerate that progress. And yeah, we just got one more question and then we can kind of wrap things up, which is sort of bringing back together both the stone fruit work and the vineyard work, and comparing it also with apple, and about compared to like the growing areas, which of those is the biggest in the US?
Oh, for apples?
For stone fruit area versus the vineyard, the grapevine area, and then also if you've got any idea about how much area is covered by apples as well.
Good question. I wish I had my apple data, because it is an important crop in the US. I mean, most of the apples are not produced in California because it's too hot. We don't have enough areas with sufficient chilling hours. I mean, I'll give you an example. I have an apple tree, a pear tree, and a quince tree in my yard, and some years, cold winters, the growing seasons following cold winters, we see those crops, but we don't see fruit on them every single year. And I'm not even in the hottest area. I live in Napa, which is closer to the coast than here in Davis, where it does tend to be hotter. So we don't have a lot of apples in California. Most of them are grown in Washington state, so 2 states north of us on the West Coast. And I don't remember the acreage off the top of my head, but it is significant. They're grown in Eastern Washington, and they're intermixed in Washington state. I've spent a fair amount of time up in Washington state for research, east of the Cascade Mountains, the big tall mountains, Mount Rainier, and the parts of the Cascade are closer to the western side of the western shore of Washington state. And east of that, in the quote unquote rain shadow, is all high desert. And there are a lot of apple orchards, cherry orchards, and these are sweet cherry orchards, and wine grape vineyards and juice grape vineyards, and more varied agriculture than we have in our wine growing regions of California, certainly.
So overall in California, is it the wine that tends to be in the northern areas, and then the stone fruit kind of can carry on throughout sort of the central areas?
No, you have them interspersed. I mean, the Central Valley of California, you have plenty of everything. Other than, I mean, almonds are so impactful on our agriculture now with over 1 million acres for us. Everything that was being replanted, whether it was peaches or pretty much any sort of crop, went into almonds or walnuts or pistachios over the past 10 years because they're just so profitable. But now, for example, with walnuts, with a glut in production, so to speak, we have, prices for walnuts to the growers are now below production costs. So that's what happens.
The swings of the market.
You can't have too much of a good thing.
Yeah, that's really interesting. I do think about Californian almonds every time you get offered almond milk in a cafe, and just thinking about the enormous production in that area.
Absolutely. Okay.
Well, thank you so much for answering all those questions, Kendra. And really fascinating insight into firstly, the sort of agricultural work going on in the USA, but also seeing things from a tree crop and a vine crop perspective, where you have these perennial problems. It's very different to the rest of our agricultural kind of pathology issues that you see. And really nice to see some sustainable approaches being taken.